So our next speaker is Professor Marcel Jaspars, who's a chair in chemistry whose research involves the discovery of new compounds with potential pharmaceutical use for marine and desert environments. His talk is entitled Microbial Agents for Marine and Desert Bacteria. Jasper, it's all yours.
Well, thank you very much for the invitation to speak here today. My presentation will be a little bit more about molecular diversity we can get from different locations and the kind of work that we've been doing. So thank you, Kate, for a wonderful introduction to some of the technologies I won't have to introduce right now. So it's really great to see what you're doing, and especially the NP linkers thing is something that I will be looking into. So just to make this work, our research center is called the Marine Biodiscovery Center, and the aim is really to discover new molecular diversity from lots of different interesting places. And the people that work there are myself as a chemist, and then Dr. Abel as a pharmacist, and Dr. Deng and Dr. Hussein are really sort of our molecular people, and then Laurent is synthetic chemist, and Russell Gray is the NMR technician. We recently lost our mass spectroscopist, so that's a bit sad. But the real reason we're doing this is to find out why nature makes these beautiful molecules. But of course, to pay the bills, we have to find applications that are suitable for industry. The technologies that we use are pretty much everything that Kate's shown already. So a lot of chromatography, spectroscopy, molecular biology, some synthesis, molecular modeling for structure determination, and software development when necessary to dereplicate molecules. The lab itself is custom built about 10 years ago right now. And despite suffering a catastrophic fire in 2014, we're able to rebuild with new equipment in 2015. As you can see, it's built for natural product isolation. We have additional facilities for microbial culture and a separate molecular biology lab over in the medical school as well. The people are typically a very international group of students. It's always very nice to have a lot of students and hosts, students and international visitors in the lab, and that works out very well in order to get to those international and interdisciplinary collaborations going. The kind of environments that we tend to look at are like Kate. We look at deep sea stuff, a lot of cold adapted environments as well, together with groups from Norway and South Africa. And as well as that, we've been working on hyper-arid desert species over the last few years as well. And we look at pretty much anything. We're very agnostic. We like to look at marine invertebrates. That's what I started out my research career with. And Rainer likes to look at fungi. I like to look at bacteria, and Wael likes to look at cyanobacteria. So it really sort of matches very well the people that we have in the lab. And the kind of things that we find are some really interesting new structural families. I'll give some examples of that in a minute. The drug applications that we find are typically sort of anti-infective, anti-cancer type activities, but the ones we found recently have been in the CNS area. So this is part of the pharmacy project that recently finished. And I'm glad to be able to report that we've just been funded for a continuation project by the European Commission. So we'll be looking for a postdoc very soon. So anybody that's in this area, yeah, just write to me and I'll make sure to advertise it in January. This kind of molecules that we found here, this is a marine fungus found from the Red Sea, was able to significantly reduce seizures in treated animals. So it's a pretty significant discovery. And secondly, the gracillins here from a sponge from the Philippines were able to reduce the characteristics associated with Alzheimer's disease in an animal model. And a group in the States has now been synthesizing a bunch of derivatives that have the pharmacophore without having any of the side effects. So again, I look forward to seeing that being published in the near future. But back to antibacterials. So the first thing I did with the Norwegians was way back in 2007 or 8, I seem to remember. I visited, and they had this species collected, which is an unusual ascidian or a sea squirt. And what it did have was, in the first extract that was taken by Margit Tedes, who was the PhD student on the project in Tromsø in Norway, was that there were 3 fractions that seemed to have high activity against a range of different bacteria. And that was exciting. And we took some time and we found some chemical structures that were quite novel. So again, it's quite rare to find novel chemical structures. And this is reminiscent of some of the antibacterials you might find. And even though the antibacterial activity is not stellar, it's quite low in fact, the chemical structure was so novel that a local company called Lytix Pharma in Tromsø bought the patent and has used that strategy of this molecule to develop derivatives that they're now working on to treat a variety of different infectious bacteria. And of course, the structure of this looks very reminiscent of linezolid. You have the same kind of core oxazolidinone in the middle. Again, these are quite rare structures in nature. The structure determination itself was quite complex, so I won't go into that today. Well, let's move on to other locations that we've looked at, and we've been fortunate to work together with some great scientists in South America, as well as some microbiologists from the UK. So again, the two people that I work with in Chile, taking the picture here, are Juana Senjo and Barbara Andrews, and they work at the University of Chile. And they really got us into this looking at desert bacteria. And I worked together with Alan Bull in Kent, and Mike Goodfellow, who is one of the renowned marine microbiologists. And again, it was just a joke film poster that we made after our first expedition to the Atacama Desert about 10 years ago. The kind of things that we find are really some unusual bacteria, and this isolate C34 has now been renamed as Streptomyces leeuwenhoekii. And again, it's a supermarket strain, as Kate referred to, strains that have a huge number of different biosynthetic gene clusters. This has something like 30 different biosynthetic gene clusters, of which we've only seen a few. And again, the first thing that Deng did on this one is to look at, for instance, the AHBA genes here, and using this kind of genome mining, basically we could see that it was different from all the ones that have been previously reported. So there must be some unknown compounds in there. What we did was grow it under different conditions. So the one strain, many compounds, or the osmotic approach. And of course, the first thing we found was not these compounds that we expected. Those that I'm reporting those first here because it makes sense. But of course, we found a whole bunch of other compounds initially. But you can see that if you put them under some nutrient limitations, you find that different compounds are produced. For instance, compounds 1 to 4, which are close to the rifampin in structure. And again, if you look at then some hospital isolates here, and again, the activity is reasonably good. But of course, rifampicin is much stronger in activity. And because the structure is so close to rifampicin and that space is so well protected with intellectual property, it's very difficult to go anywhere with this. We also found a bunch of other compounds, for instance, the chaxolactins here, some siderophores, some hygromycins, which were produced in response to some chemical cues that we put into the system. So again, all kinds of approaches to find the compounds that are out there according to the genome. And again, these antibacterial activities are not great, but they're not bad. These compounds were found by a German group to be much more active against cancer. So again, I'm not sure where they are with that at the moment. So again, using interesting strains, you often find that the genomes are unusual. And you find in this case, the gene cluster, again, 39 predicted biosynthetic gene clusters is quite high. And you can see again the families that they come from. And one prediction was that it contained a LASU peptide. LASU peptides are interesting because we really don't know what they're there for. But chemically, they're unusual, but they're becoming a nuisance. We see a lot of these, and we have to find them very quickly to dereplicate them because we don't really want to isolate another LASU peptide. Now we find quite a few. The problem has always been in identifying that you have that sequence because under normal conditions, this won't fragment. So technically, what we normally get is this. You have a compound with a loop and a string, essentially, and the fragmentation will take place all the way down to this point and leave you the cycle, the macrocycle behind, which is okay. You can show that at least there is that sequence there, but it would be great to go further than that. So Scott Yarmush, one of our PhD students, was very keen to find a different technology. And what we did was we used this high energy collision approach in one of the systems. Actually, we had to go to Germany to find a system that had this ability. What you then get is it fragments the entire system, including you get these T-shaped fragments, which are not so useful, but then you also get some linear fragments, which allow you to do a full sequence elucidation of the structure that you have. And of course, the first thing we did is to show that on an isolated peptide. And you can show that that sequence is indeed there. You can't prove that it's a Lasso peptide in three-dimensional structure, but what you can do is prove that the entire sequence is there and to replicate for that. And we tried that on in fact, we tried it on an extract as well. So what you see here is the whole genome. You see the extract, which has a compound with a molecular weight that is consistent with one of the biosynthetic gene clusters. And that again, by fragmenting it in the extract, so by taking essentially that extracted ion current and fragmenting that particular mass, what we're able to show is that this was citralacin C, which was a known compound, a known Lasso peptide. So rather than having to isolate it and verify its structure using this technique, we're able to do that in the extract. And that's really a very useful advance for us to dereplicate rather than to have to find the same compound over and over again. I just wanted to talk a little bit about this, the idea of spinning out some of the technology that we've developed. And this is a different technology, but it's been an enjoyable experience. So most of the company's focus will be on cancer, but the technology is equally applicable to creating new chemical diversity for antibacterial agents. And again, company was really formed 2015 to start with. It was called Riptide, and we won some awards, which was great. But of course, what they hate the most when you win these awards is that you fail quickly. We failed in 2017, but we started again, and we're now called Gyrox Therapeutics. Here's Orode, our first researcher, planting a tree, because it's Oxford's approach right now is if you start a new company, you plant a tree in a forest somewhere. That was a nice occasion. What the company does essentially is to try and use a chemoenzymatic approach. So what we do is we use substrates that are made of not natural and unnatural amino acids, and you take that and you allow the enzymes that we are isolated from a series of marine organisms to put in heterocycles, in this case thiazolines and oxazolines, which can be oxidized as well to thiols and oxazoles. You can do some macrocyclization to generate a complex modified macrocyclic peptide, and that allows you then to use other enzymes to oxidize the heterocycles from thiazoline to thiazole, oxazoline to oxazole, and also to add prenyl groups to reduce the polarity. And because of the possibility of including chemical handles, we're able to add some interesting new chemistry as well. So if you put an azide there, you can do some click chemistry to add groups that allow you to see where the molecule is in biological system. Finally, you can use that to take a lot of different, the same substrate with different enzyme cocktails and different chemistry to generate a whole bunch of biological or chemical diversity that you might want to test. So again, you can make families of molecules very quickly. So where we're at the moment with the company is that we've been able to start automating this whole process to make hundreds of compounds at a time, and starting with a design process. So what we're doing right now is using design molecules to fit into target sites that can then hit those targets. And we make a family of, say, 80 to 100 molecules to make sure that we cover that entire target site with design molecules that we have and see what the activity is. So I'll let you know what happens to that in a few years' time. I'd like to show you some of the kind of diversity that we can generate. And again, you can see that these molecules have either just normal amino acids with some unusual amino acids in there. They can have sugars, they can have fatty chains, they can have the heterocycles. And again, this is just the beginning. This was 3 years ago. And now we're getting to much more complex chemical structures within the company. Finally, I'd like to thank everybody that was involved. Margie in particular for synoxazolidinone, Samaya and Mustafa for the traxapeptin. The huascopeptins was Carlos and Scott. YL, particularly for the cyclic peptide work, starting it off, and then of course the current people working in the company, Gyrox. None of this would have been possible without the financial input of a lot of different funders, and I mentioned the EU already today, but particularly like to point out the Leverhulme Trust as being one of my favorite funding sources because of the way they fund high-risk research. So thank you very much.
Thank you very much for your talk, Marcel.